Hi everyone, we are continuing our survey of the biological molecules. By point of review, we've just come from talking about the carbohydrates, and we've also proceeded then with talking about the lipids. And if you recall, carbohydrates are anything with the formula CH2O. So for every carbon there are 2 hydrogens and 1 oxygen, and there can be a variety of carbons. So we put N here to refer to, it depends on how many carbons there are. But let's say if there were 5 carbons, you'd have 12 hydrogens and 5 oxygens. If there were 6 carbons, you'd have 12 hydrogens and 6 oxygens. Okay. Carbohydrates, they're chiefly known for their ability as they serve as short-term energy storage. So when I eat my breakfast in the morning, I'll probably consume those carbohydrates and they'll be held as carbohydrates for a while and then perhaps broken down into energy. And if I do not use that energy that's consumed in the carbohydrates, then it gets stored as a long-term storage, and that's what the lipids are mostly known for. So short-term energy storage and long-term energy storage would be the lipids. Remember, lipids are generally made out of hydrocarbons, which are long chains of carbon that are completely full of hydrogen. Okay. Proceeding on then with proteins. Proteins are a polymer just like the carbohydrates and lipids, but instead of using saccharides or fatty acids, as in the case of carbohydrates and lipids, these have a monomer which is called an amino acid. And if you add a bunch of amino acids into a long chain, we call it a protein. Okay. Now, how proteins work is that the amino acids that are used, there's 20 different ones, and because there's 20 different ones, some can have varying chemistry with one another. So, for example, this particular amino acid here might be attracted to this one, and this one over here might be attracted to this one. Okay, so because they have this variation in chemistry, if you let this chain kind of go, they'll automatically fold up into a shape, a three-dimensional shape. And for proteins, it's important to remember that the shape will determine the function of the protein. Okay, and this is just a small example, but in reality, these amino acid chains can be quite long, and when they fold up, they can fold up into pretty complex structures. Proteins have a variety of functions. The first is that they help regulate chemical reactions. The class of proteins that does this are known as enzymes. And for example, an enzyme looks something like this. You have basically some sort of substance, which is called a substrate, and then you have the enzyme. Now the enzyme is going to have what's known as an active site. So that's this little spot that I'm drawing here. And the active site is going to have a shape that fits the substrate. So in this example, maltose, which is a disaccharide, has this particular shape, and that fits nicely into this little shape of the active site of the enzyme. So enzymes are usually pretty specific. They have a certain substrate to which they can bind, and they bind only to that particular substrate. And the way that we usually name an enzyme is that their name is based upon the substrate to which they bind. So for example, this particular one has the active site which will bind maltose, so we would call this enzyme maltase. Okay, the enzymes generally have the last 3 letters as ACE, and then it's just whatever it binds, and then you change the last letters to ACE to signify that it's the enzyme of that particular substrate. Okay, now in either case, the enzyme will help, in this example, break maltose down into 2 individual monomers of glucose. Okay, now you might be at this point be thinking, well, wait a minute, I thought that this was hydrolysis. You know, if you're paying real close attention, you're seeing that this is actually a chemical reaction that we've talked about. You take some sort of polymer, you add some water to it, and it breaks down into its individual monomers, which in this case is glucose. And this would certainly be an example of hydrolysis. And you know, the contrary would be dehydration synthesis. And that is true, and hydrolysis will occur on its own without the presence of an enzyme. But if you add some enzyme in there, it actually makes this whole reaction occur much faster, probably 1,000 to 10,000 to 100,000 times faster. So in that sense, enzymes can be a pretty nice tool that the cell can use to speed up stuff or slow stuff down, depending on what it wants to do. We'll learn more about these enzymes a little bit later in the course. Another thing that proteins do is they provide structural support. So oftentimes proteins will bind with carbohydrates or lipids to kind of keep things stuck together. And a perfect example of this is that inside the cell there's a whole system of tubes and rods and spindles known as the cytoskeleton, which give the individual cells their shape. So instead of your cell just being kind of a squishy thing of fat and water, it actually has a little bit of more of a solid shape or structure to it, and that's due to those proteins. There's a whole bunch of other examples where you might see proteins giving some structure to something. Proteins also really help with what's known as transport. And when we say transport, one of the things that comes to mind is the cell membrane. Okay, recall that the cell membrane is made up of phospholipids. And so because it's made up of phospholipids, again, phospholipids are these cute little molecules that have a polar head and a non-polar tail, which is made out of fatty acids. And because the non-polar stuff likes to associate with the non-polar stuff from both the layers, you get a nice hydrophobic region inside the middle of the membrane. Okay, and if you have something that is hydrophilic, which is out here outside of the cell where there's a bunch of water, then that hydrophilic thing has a hard time making it through the hydrophobic part of the membrane. So a workaround is that there's actually these channels inside the membrane into which something that's hydrophilic could pass, and then likewise something inside the cell could pass out. So when we say transport, this is kind of one of the things that we're talking about, that stuff can get into and out of the cell. Another function is chemical signaling, and when we're talking about that, the particular term we use is hormones. Hormones are things that are secreted by one part of the body or one part of the organism, and they can travel to a different part of the organism to exert some sort of effect. So, for example, my pancreas can release a hormone known as insulin, and that insulin can go out into the rest of my body and tell all of the cells in my body to uptake glucose into the cells, so to take sugar out of my blood and put it into their cells. So that's one example there. And then lastly here, they can also be involved in defense. When we're talking about this, we usually come up with, we're talking about these things, antibodies. Antibodies are like little seek and destroy substances which can attach and destroy very specific things, like a very specific strain of bacteria or a very specific virus or whatnot. All right, so those are the different things that proteins do. Let's talk a little bit about what they're made out of. We said they're made out of amino acids, and there are a general structure of an amino acid looks something like this. You have a carbon atom, and then on one side of the amino acid you have a carboxylic functional group, and on the other side you have this group, which, can you tell me which this group is? This is the amino group. And that's fitting because we call these amino acids, okay? Then all of the amino acids, so they're going to have an amino group and a carboxylic group, and then they're going to have a unique side chain which has a special or some sort of special chemical property to it that makes it a certain way, okay? So there are 20 different types of amino acids. They look like this. So all the proteins in your body are made out of 20 different types of amino acids. And in the same way that out of, in the English alphabet we have, I think, 26 letters, and out of those 26 letters you can make almost an infinite array of words. So in the body, out of the 20 amino acids, can we make a nearly infinite array of proteins? Okay. So here they are. You've got glycine, alanine, valine, leucine, methionine, isoleucine, serine, threonine, cysteine, proline, asparagine, glutamine, lysine, arginine, histidine, aspartate, glutamate, phenylalanine, tyrosine, and tryptophan. And this was a biochemistry class, you may have to learn how to draw all of these, but you certainly don't have to learn how to do that in this class. And I'm not even going to ask you to memorize all of these amino acids. But if you really wanted to, you could memorize this one, which is the simplest of all, glycine, just so you could boast about knowing the structure of one amino acid. Its side chain is just a very simple H, and that's it. All right, so those are the different amino acids out there. And what happens is when amino acids bond together, this is what occurs. You have the hydroxyl of one carboxylic group, okay, and the hydrogen of one of the adjacent, the amino acids of one of the adjacent, or the amino groups of one of the adjacent amino acids are attaching together, okay. So this and this, boom, boom, they come together and they form this interesting structure was called a peptide bond. And there's going to be, depending on how many amino acids, a whole bunch of these peptide bonds. And so sometimes when you have an amino acid chain that's formed of many, many amino acids all bound together by peptide bonds, sometimes the name that we use is we call it a polypeptide. And the polypeptide is kind of like another name for a protein, but it generally refers to more of a primitive state of the protein, being the long chain of amino acids before they undergo any sort of shape change. All right, so proteins have a kind of a complex hierarchy of structure to them. The first is that they start off with just a primary structure, which is just nothing more than a link of many amino acids. Now we said that some of these amino acids are going to have a variety of different types of properties to them, which is going to give them kind of a unique assortment of chemical attractions. So like this one might be this one, this one might be attracted to that one, and this one might be attracted to that one, and so on and so forth. Well, based upon these different chemical attractions that the amino acids can have, they can form different structures, which will get us to the secondary structure. Before we get there, though, I just want to point out an example of a primary structure. This is what insulin looks like. And you can see it's just a long chain of one type of amino acids, and then there's another long chain, and the two chains are attached together. It's a very simple protein. Now, the secondary structure is when we get the formation of these two characteristic shapes. The alpha helix, which is kind of like you can think of this being kind of like a coil shape, and the beta pleated sheets, which are just kind of like a more rigid series of folds. So when this long chain of amino acids starts to fold, there's a certain pattern that we see where the amino acids will actually start to coil around one another and form this type of shape. And then sometimes the amino acids just kind of scrunch together, forming the pleated sheet. So these two things will start to form. If you wanted to kind of see a little bit of an example here, here's the kind of the chain of amino acids, which is starting to kind of swirl into a coil here. On another part of the chain of amino acids, you had the kind of the pleated linking of the amino acids, and you can kind of see this is kind of the molecular scheme showing how this chemistry occurs. Generally, the alpha helix is going to be, well, both of them are usually formed because of hydrogen bonds. Okay, so that's what keeps them together. And then, furthermore, after the alpha helix and the beta pleated sheets have formed, then the whole long polypeptide, the whole long chain of amino acids can then develop more of a three-dimensional shape known as the tertiary structure. So, for example, if I go back to this picture, once we have the alpha helix and the beta pleated sheet, perhaps one part of this protein would be attached to like this part or whatnot. So the whole long chain of amino acids is developing a highly complex three-dimensional shape. That's known as the tertiary structure. There's a couple different things that would make the different parts of the protein bind with one another. It usually has to deal with the side chains of the amino acid groups. So one thing that can happen between side chains is ionic bond. You could have hydrogen bonds happening between the side chains of one amino acid and another. You could have what's called a disulfide linkage happening between one side chain of an amino acid and another, or just hydrophobic interactions. You've got two nonpolar substances that want to be next to each other. And so at this point, I always like to stop and just think about how crazy this is, that all these extraordinarily complex proteins like hemoglobin, the stuff that carries oxygen in your blood, or myosin, the thing that binds your muscles, or even the pigment in your eyes that gives it a color. All of these millions of different types of proteins, they do what they do because they have this crazy complex way of binding that's determined specifically by the shape that's formed due to the sequence, the specific sequence of amino acids that are forming the protein. So lots of complexity there. You're starting with a simple chain of amino acids, and the body kind of knows how to arrange amino acids in order to make the type of shape of protein that's needed to do something that's super highly specialized. Real quick, just FYI, if you have curly hair, the reason you have curly hair is because there's a lot of these disulfide linkages which link the proteins together to form kind of a curly structure in your hair. And if you take a shower, it breaks these momentarily so that your hair is straight, but then once it dries again, it's curly again because those linkages have reformed. Okay, and then lastly, you can have one tertiary structure of one protein and another tertiary structure of another one, and they can come together to form what's called a quaternary protein. So a quaternary protein is going to be one or two or more amino acid chains associating with one another. Here's a couple examples. This is myosin. This is the thing that makes your muscles move, and you can see that there's a complex array of alpha helices and beta pleated sheets would be here, and then it's all folded in on itself, so that's the tertiary structure. And then we can even see we've got two different chains of amino acids that are all associating. So they're indicated by the different colors here. So this is actually a quaternary structure. Another example is hemoglobin. The specific shape and arrangement of these amino acids is such that it allows the attachment of four what are called heme groups. And heme groups are these little molecular structures that have iron in them, which are attracted to oxygen. So hemoglobin is the thing that can kind of carry oxygen in your blood. And you don't have to know that. That's just kind of FYI. One last bit here about proteins before we proceed. We said that the shape is what determines the function of a protein. So what do you suppose happens when a protein loses its shape? Well, it loses its function. And that's what we mean by this word denaturing. So to denature a protein is to say that the protein has lost its shape, and consequently its function is going to be changed. This can happen for a variety of reasons. It could be a change in the pH. It could be, that's what this little symbol means here. It means change. Change in the pH could be a change in the temperature. Could be a change in the pressure. Could be a change in the what's called the osmolarity, the saltiness of something. So a variety of different things. You don't have to know these specific stuff, but just know that there's a variety of things that can change the shape of protein. And this is the very reason why we need to maintain a constant environment inside of our cells. We have to remain what's called in homeostasis. And homeostasis, if you recall from the first chapter, means that we have a constant condition in our body. And that's important because if the pH sways too much, or the temperature changes too much, or the pressure, the osmolarity, any of these things, if they change too much inside of our bodies, then the proteins inside of our cells are going to start to lose their shape and not function anymore. Kind of an interesting example of this is if you ever go to the gym and you're lifting heavy weights, there comes a point at which your muscles are so tired they don't even work anymore. You can't really even move the weight up and down. And what's happening there is as you work out, so there's some acid that's produced in your muscles, and that acid is going to increase the acidity, so it's going to lower the pH, and then suddenly there comes a point at which the proteins in your muscles, they don't function anymore, and so you have to kind of take a break. The pH the acid will clear out, and then your muscles will function again, but sometimes it can be a little painful, and it can take a little while. Another example is this. All right, so this is an egg white right here, and normally if you crack an egg and look at the egg white, it's clear. There's a protein in here called albumin, spelled like this, and when albumin is heated up, it denatures, and so albumin in this example allows light to pass through, but once it's heated up, it does not allow light to pass through because the shape is changed such where it does not let light pass, but instead reflects light, so it appears a white color like this, and it's more stiff and hard. So even there, in a simple example of cooking eggs, are you experiencing the process of denaturing. Alright, so here's a review question for you. What elements are proteins made out of? Give at least 3 examples of types of proteins and their functions. What is the monomer that makes up proteins, and what are the proteins? Why are proteins often called polypeptides? Review the 4 levels of protein structure. Draw and describe each. If one amino acid is changed in a protein, why might this change the function of that protein? And then lastly, what does it mean if a protein has been denatured? Well, go ahead and pause the video and take some time to work through these and see if you can. All right, part A, what elements are proteins made of? Well, remember it starts with a carbon atom. And on one side of the amino acid, there's a carboxylic group. And on the other side, there is a, oops, I already messed this up. There is an amino group. So then your answer would be that they've made out of hydrogen, carbons, oxygen, and then this is kind of interesting, nitrogen. There's not a lot of nitrogen in fats or carbohydrates, so that's kind of an interesting characteristic of proteins. There's quite a bit of nitrogen and nucleic acids as well, but so keep that in mind. All right, 3 examples of types of proteins and their functions. We could have something like an enzyme. Enzyme is going to either speed up or slow down a chemical reaction. You could have something like a hormone, which is a chemical signaling molecule. Example of these would be maltase, like we said earlier, or even like lactase. That would be the enzyme which breaks down lactose. The hormone, an example would be insulin, like we mentioned. And then another type of protein could be something like a antibody, which is used for defense, or something like actin, which is going to be the thing that forms the cytoskeleton of the cell. Okay, so some examples there. The monomer that makes up proteins is an amino acid. Proteins are often called polypeptides because of the fact that in between one amino acid and another, there's a special bond called a peptide bond. And when you have a bunch of peptide bonds together, you can call it a polypeptide. The 4 levels of protein structure are the following. You have the primary, and this is just going to be a, I'll draw it over here. This is just going to be a chain of amino acids. You have the secondary, and this means that's the chain of amino acids has started to fold based on the chemistry. So now you're having the emergence of the alpha helices and beta pleated sheets. Okay. Then you have the tertiary, and this is going to be the protein is starting to develop a 3-dimensional shape. The entire polypeptide, that is, not just small regions of it, which are turning into alpha helices and beta pleated sheets, but the whole thing is going to start to have a 3-dimensional shape, and this is because of the side chain interactions of amino acids. Okay, and then we have, lastly, the quaternary. This means that there are several polypeptides associated with one another. If you care to abbreviate these, oftentimes the abbreviation for primary, secondary, tertiary, and quaternary is the following. It's the number with this little circle on it. So that would mean primary. This means secondary. This means tertiary. And this would mean quaternary. So you're welcome on an exam or something like that to abbreviate those. If you change the amino acid of one protein, why might this change the function of that protein? Well, the shape is determined by the series of amino acids, and so if you alter one amino acid, it can cause a slight change in the shape, and we said it's the shape that determines the function, so changing the shape would lead to a change in the function of the protein. And what does it mean if protein has been denatured? It means that the shape has been changed, either due to temperature, pH, osmolarity, pressure. All right, then on to our last category of the biological molecules, which is the nucleic acids. The nucleic acids are made out of things called nucleotides. So this is the monomer that makes up nucleic acids. Here is what a nucleotide looks like. So again, this is polymer, but each one of these little things in the polymer is a monomer, and in our example of the DNA, that is nucleotides. Here's an example of a series of nucleotides. Each thing that you see here is one nucleotide. So there is one nucleotide. Here is another nucleotide. There's another nucleotide. Okay. And if you look at this, you're going to start to see that each nucleotide has 3 parts to it. There is, and I bet you if I just asked you, you could probably spot the parts. But the first is that there's going to be this thing, and I'm hoping that you all know what that is. That is a phosphate group. And it's the phosphate group that kind of attaches all, it's the thing that kind of forms the bridge to which all of the nucleotides are bound. So this is sometimes referred to as the phosphate backbone. So it can form kind of the backbone of the molecule. Nucleic acids such as DNA can be a really long molecule, and so they kind of need a little bit of a backbone to give them some structure. The second group then is going to be this thing here. Can you determine what type of molecule this is? Have you seen something that looks like this before? That is going to be a sugar. And particularly it's a pentose sugar, so 5 carbons. And there's a variety of different types of sugars that you might see in nucleic acids. Actually, I shouldn't say a variety, there's really only 2. And depending on what sugar it is, determines which type of nucleic acid it is. Oh, excuse me. So we have kind of 2 types of nucleic acids that we talk a lot about, DNA and RNA. You're probably familiar with DNA and a little bit less familiar with RNA. Well, DNA has this sugar that's called deoxyribose, and RNA has this sugar which is called ribose. Deoxyribose and ribose. So depending on which one of these sugars is present, if it's deoxyribose, then this is likely a strand of DNA. If it's ribose, then this is likely a strand of RNA. And here's the difference between those. And you're going to be probably a little bit startled to even see that there is a difference because they look identical from first glance. But if you look really carefully, you'll see there is a very slight difference. Right here, there is an oxygen, and right here, there is not. So that's why we call it deoxy, because one of the oxygens is kind of being deoxified, if you will. Okay. We'll talk a little bit more about the difference between DNA and RNA a little bit later. Another thing then that we find on the nucleotides is this last bit here. So these little bits here, these are pretty important. These are called the nitrogenous bases. And nitrogenous bases can be characterized into 2 categories. There are the pyrimidines, and there are the purines. The pyrimidines are the C's and the G's, and the purines are the, oh, I already made a mistake. C's and the T's. The purines are the A's and the G's. And if you take a look, you can see that the C's and the T's, they just have 1 ring. whereas the G's and the A's, they have 2 rings. Okay, so that's what's different between them. Pyrimidines always pair up with purines on a double strand of nucleic acid, and I'll show you that in a minute. One exception to all of this, when we're talking about DNA, this is the case. When we're talking about RNA, another difference that RNA has is that the T is actually a U. Okay. What these letters stand for is the actual name of the compound. So C stands for cytosine, T stands for thymine, U stands for uracil, A stands for adenine, and G stands for guanine. But you can just refer to them as C, T, U, and A, and G. And it's the arrangement of these 4 little letters that stores all of the information required to build your entire body. So it's at this point I like to pause and just think about that for a minute. You have 4 letters, and DNA is a huge molecule. You arrange these 4 letters in a whole bunch of different combinations, and it's nothing but the mere arrangement of 4 of these types of molecules which will tell your body how to make an infinite array of proteins, and from that infinite array of proteins you develop the extraordinarily complex structure that you have. This will never cease to amaze me. It's almost so amazing it's hard to even believe it's true. And if you don't think of it in that way, maybe just sit by a tree down by the river someday and just think about nucleotides. All right, moving on. DNA versus RNA. There's a good chart here that kind of sums up some of the big differences between these, and you're going to want to be able to spot these differences on the exam. Okay. The first is the function. DNA is going to be the thing that carries most of the genetic information, whereas RNA is involved more in protein synthesis, so it makes proteins. Ooh, excuse me. Tired today, I guess. The location of these is that DNA is found in the nucleus of the cell and it stays there. It's a huge molecule and it doesn't really move all that much, whereas RNA can leave the nucleus. Okay, so here's a picture of a cell. Here is the nucleus. Well, you can think of DNA, which is the one that's found in the nucleus, as being kind of like the blueprints to building stuff, and then the stuff that's actually built is proteins. Okay, and out in the cell there's a whole bunch of these special organelles which are called ribosomes, and the ribosomes are the places where protein is made. And we're kind of skipping ahead to chapter 4, but this will be a good intro to it. And how it works is that you have all of the information, the blueprints for how to build the proteins in the nucleus, but that information is stored on such a huge molecule it can't make it out to the place where the proteins are actually built or assembled. So little snippets, little copies of the information stored on DNA need to be made in order and then launched over to the place where protein is made. So that's where we get this type of RNA called messenger RNA. It comes onto the scene. And that messenger RNA is going to leave the nucleus. It's basically just a copy of a small piece of the DNA. And the messenger RNA can then make it out to the ribosomes, where then the instructions can be utilized and turned into an actual product, which is the protein. Okay. We'll talk a whole bunch more about that process in a minute, and also later on in the course. The structure, DNA has the shape of what's called a double helix, whereas RNA is usually single-stranded. I'll tell you what I mean by double helix in just a minute. Sugar, we already said this, DNA's got this sugar called deoxyribose, whereas RNA has a sugar called ribose. And then the pyrimidines are going to be cysteine and thymine, and then, cytosine and thymine, and also cytosine and uracil. So in DNA and RNA, they're a little bit different because RNA does not have thymine. Instead, it has uracil, whereas the purines are exactly the same between the two groups. Okay. All right, so now looking at DNA. DNA is this shape we call the double helix. And to understand a double helix, imagine kind of a ladder. A ladder's got 2 rungs to it, right? Or it's got 2 kind of supports to it, and then there's a bunch of rungs in the middle. Well, the rungs in the middle, if we're talking about DNA, this is where we find the purines and the pyrimidines, the nitrogenous bases. And so you might have one that would be like CAATGC, let's say. And then on the other side, you're going to have what are called complementary matched base pairs. So there's this rule called complementary base pairing, where C's always attached to G's and A's always attached to T's. It's just the nature of the chemistry between them. Okay, so if this was one run of the ladder and this was the other, the bases in the middle here would be G, because C always goes with G, T, because A always goes with T, another T, T and A always go together, so on this next one it would be A, C, and G. Alright, and then these two sides of the ladder are usually bonded together with hydrogen bonds. Now if you have this ladder then, and you twist it, then you have the shape of the double helix. So now you're starting to see kind of this thing. It's really just, think about it as being a twisted ladder, and then each of these little rungs of the ladder are a base pair of nucleotides, or a base pair of a pair of nitrogenous bases. Okay. If you wanted to see kind of how the rungs of the ladder are bound together, here's kind of a diagram that shows you. Again, this would be the phosphate backbone, and if we're talking about double-stranded nucleic acids such as DNA, you're going to have one backbone and another backbone, and then the two strands are bound together through these, which are the hydrogen bonds. And a pyrimidine such as thymine always attaches to a purine, adenine, and the purine guanine always attaches to the pyrimidine, cytosine, and vice versa. Okay. One last bit here about RNA, and then we'll be done. RNA, there's a whole bunch of different types to it, and I already introduced you to messenger RNA. This is the thing that takes the information of information stored in the nucleus and brings it out to the site where proteins are made. But the process of making protein is also going to utilize these other types. And I'll just make a few notes about them. Okay, so messenger RNA, we said, is so a way to think about this is it brings the plans, or it brings the information, the blueprints, to the site of protein production, which is the ribosome in the side of the cell. The ribosomal RNA, this is what actually makes or forms the ribosome, which is the thing that makes protein. Transfer RNA is going to be kind of a special molecule because it's really the link between nucleic acids and protein. So the way that a nucleic acid can be used to make a protein is heavily dependent upon the use of these molecules called transfer RNA. Transfer RNA has one side of it that binds to nucleic acid and another side of it that binds to amino acids. And so as the information of DNA is passing through the ribosome, it can figure out what amino acids it needs to bring and attach those to make a big long chain of amino acids. I'll show you a picture of that in a second. And then there's microRNA. This stuff was kind of more recently discovered. It's a little less well understood, but it's basically something that can regulate which proteins are made and not made, because it can bind to stuff and shut genes off and turn off protein production or turn it on. Alright, so here's kind of a picture that shows the whole long process. This is a really important process called translation, and we'll talk probably a whole chapter about it later in this semester, but for now I just want to introduce kind of a brief picture here. What you're seeing is this is a ribosome. Okay, this is the place where protein is made. And out of that ribosome, you're seeing a growing chain of peptides emerging. So, okay, this is going to be a polypeptide, which will, once it leaves, turn into a protein. Now, the way that this chain is made is that this molecule, messenger RNA, which is basically a copy of DNA, which is small enough so it can leave the nucleus of the cell, it is going to be fed through this ribosome. And as it's fed through the ribosome, you have a whole series of transfer RNA molecules, these things here, which will recognize and bind to these different, they're called codons. They're basically a series of 3 nucleotides, okay, 3 nitrogenous bases. So for each one of these series of 3 nitrogenous bases, one transfer RNA is going to be bound to it. Now, each one of these transfer RNAs, when it's attaching to here, is going to have with it an amino acid. So when you look at this little thing here, let's say this is the transfer RNA. It's bound to the messenger RNA, and it's also carrying with it an amino acid. And then as this continues to move through it, the amino acid gets linked onto the chain, so then it gets one bigger. And then as this continues to feed through, another amino acid or another transfer RNA will come, and another amino acid will be added, and then it just keeps proceeding. In no time at all, you have a pretty long amino acid chain popping out of the ribosome, and then it again will fold into a protein and go do its thing. So those are kind of the different types of RNA. Again, these are more involved in making proteins, whereas DNA is more just kind of the database. It's kind of the place where the information is stored. Right, our final review question. What are the elements that nucleic acids are made out of? Give 2 examples of a nucleic acid and its main function in the cell. What is the monomer that makes up nucleic acids? What are 3 parts of one nucleotide? Describe how nucleotides form the DNA double helix structure. Go ahead and pause the video and see if you can work through these, and I'll tell you what the answers are. Alright, let's take a look at part A. For starters, what elements are nucleic acids made out of? Well, they have phosphate groups on them, so there's going to be quite a bit of phosphorus, and phosphorus is one of the things that makes nucleic acids a little bit more unique. Nucleic acids have a lot of phosphorus in them. You know, there's going to be phosphorus and phospholipids too, but it's kind of phospholipids and nucleic acids are the things that have phosphorus in it. Then, of course, they're going to have some carbon in them. And then a whole bunch of nitrogen actually too, because they have those nitrogenous bases, and then probably some oxygen and hydrogen. Okay, so it's the presence of nitrogen and phosphorus which was going to make the nucleic acids a little bit unique. Examples of nucleic acid and the main function in the cell: we've got DNA and we've got RNA. DNA stores info. Stores information. RNA helps make proteins. The monomer that makes up nucleic acids would be a nucleotide. Okay, this thing here. And the parts of the nucleotide, we remember we've got a phosphate group. We've got a sugar. Pentose sugar, and depending on what the sugar is, determines what type of nucleic acid we're talking about. And then lastly, a nitrogenous base, of which there are the pyrimidines and the purines. Purines are A and G, pyrimidines are C and T. Alright, describe how nucleotides form the DNA double helix. Well, basically you've got 2 chains. There are 2 kind of lengths, and then there's a whole bunch of nitrogenous bases. And the nitrogenous bases of one long chain are attracted to the nitrogenous series of nitrogenous bases on the other, and this is due to hydrogen bonding. Okay, so a C would be attached to a G. G's are always attached to C, and then A is always attached to T's. Okay, so that's how the chains are formed, and then it's going to bend in a circular fashion to form the double helix. And this will conclude our Chapter 3 on biological molecules.